Hi, my name is Miriam Herrera, and my project is called Creating a Transgenic Tool to Knock Out the unc-18 gene. As an overview, I'm going to be giving background on my project as well as my project goal or what we're trying to achieve, the methods for how I achieve this, as well as next steps going forward and the implications of my work. First thing I'm going to tell you about is neurons. Neurons are nerve cells within the brain and other parts of the body, but mainly the brain. They rely on both electrical and chemical communication to transmit sensory information and influence behavior, but we're going to focus on only chemical communication. Chemical communication occurs at the synapse, which is the space between two neurons, and it relies on the release of neurotransmitters, which are basically chemical signals that are sent throughout the body. Neurotransmitters are held within vesicles and released using vesicle docking, which is when vesicles dock or they fuse to the presynaptic membrane, which is the membrane of the neuron, and open up and release into the synapse. This is a figure representation of chemical communication, showing two neurons and the synapse being the space between them, where vesicles have already docked into the presynaptic membrane, the membrane of the neuron, and are releasing these neurotransmitters into the synapse. The model for my project is C. elegans nematodes, which is basically a type of nematode widely used as a model organism within all types of biological research due to the fact that it is easy to maintain and it has a fully mapped genome. The unc-18 gene is a gene within C. elegans nematodes involved in vesicle docking and neurotransmitter release, specifically in head and ventral cord neurons of the organisms. This is a visual representation of C. elegans nematodes, specifically a hermaphrodite here, onto the Cre-Lox system. So the Cre-Lox system is a genetic mechanism that can be used to knock out genes by targeting them using Cre recombinase, the enzyme, as well as LOXP sites. Knocking out a gene basically means turning it off or silencing it, just keeping it from being expressed. And the way to do that using the Cre-Lox system is through gene inversion, which is basically flipping around a gene so that it's backwards within the genome. The Cre-Lox system is very specific in worms, well, as specific as you want it to be since it's based off of where you put LOXP sites. To give you a better idea of what a LOXP site is, think of it like this. A LOXP site is a specific piece of DNA, a sequence, that is basically used as a flag. So you need two of these flags to surround a piece of DNA to sort of point out to the enzyme Cre recombinase, hey, this is where we are, this is the boundaries you need to stay between, come here and cut in this area. And basically you need two of these pieces to form one set. You need two sets to completely flip a gene rather than just cutting it out. So the goal of my project is to make a tool to knock out or silence the unc-18 gene within C. elegans nematodes, and the way we will do this is by introducing LOXP sites around the unc-18 gene. To achieve this goal, I had to use two separate sets of methods, and this is my first set, DNA conformation, which was basically done to ensure the correct genomic contents. To preface this, I will say that there was a pre-existing worm line, so a family of worms that contained the first LOXP set already. So there was already one set, or was supposed to be one set of LOXP sites around the unc-18 gene already. So this part of the methods is me just confirming that this is true for us to do the steps afterward. So to do this, I had to perform a worm lysis, which basically requires that I kill the worms and extract their DNA so that I can use it in a PCR. A PCR is a polymerase chain reaction, which is basically a mechanism used to replicate a ton of copies of DNA so that we have a bunch of it and can use it for sequencing or replication or for putting it into something else. So in this situation, we needed a bunch of copies of this DNA so that we could sequence it to basically figure out, what specific nucleotides made up the sequence, what this actually was that we had taken from the worms and extracted, to see if it actually matched the sequence of the first LOXP set, whether or not it actually was. So the specific type of sequencing we had to do to specifically find out if the first LOXP site set was there is Sanger sequencing. And so here's a visual representation just to make it easier to understand. It's similar to PCR in the sense that a bunch of copies of DNA are being made and replicated over time, over and over and over, using nucleotides. But also, Sanger sequencing is different because it has the presence of dideoxynucleotides, which are basically nucleotides that are color-coded based on specific nucleotides. So here in the picture you can see that T nucleotides are pink, A nucleotides are blue, G nucleotides are yellow, and C nucleotides are green. Basically, as soon as one of these nucleotides was integrated into a copy of the original DNA sequence, it cut off the sequence entirely so that no more nucleotides could be added on. So by the end of this similar thing to PCR, there would be a bunch of different sizes of this one DNA sequence that when you stacked up would color code the specific nucleotides in order so that when we shined it under a light, we could tell what the sequence was. After sending that DNA out for sequencing, it did come back that it was the first set of LOXP sites, which allowed me to go on to my second set of methods, which was molecular cloning, isolating LOXP sites and an mCherry biomarker. So basically, to do this, we had plasmid DNA, which is a circular piece of DNA that contained a second set of LOXP sites, as well as a backwards-facing mCherry biomarker. We already know what the second LOXP set is useful for. Once we put it in, it'll actually help the Cre recombinase target and invert the target gene unc-18. But mCherry is a fluorescent biomarker. So basically, when you activate it, when it gets expressed within an organism, it glows red, but because it's backwards, if we inserted it right now, it wouldn't work. So basically, we have this plasmid DNA, and I put it through another PCR so that I could get a bunch of copies of this DNA since we need it to put into the worms. And once I had all these copies from running the PCR, I put it through a gel electrophoresis, basically to separate the parts of the DNA based on size so that I could cut out the specific piece that I needed with only the second set of LOXP sites as well as the mCherry biomarker. For next steps in future work for this project, the DNA will be inserted into the worm genome, which will result in the cutting of 2 LOXP sites and then glowing neurons. So for DNA insertion, the DNA tool containing the backwards mCherry and the second set of LOXP sites will be inserted into the worm genome using CRISPR-Cas9 after it's injected into the worms. I didn't have clearance to inject the worms myself, so someone within my lab is doing that now. But once this is inside the worms, it will trigger the tool, which will result in the cutting of the 2 LOXP sites, since they will signal to Cre recombinase, hey, I'm here. So it'll cut and invert the entire unc-18 gene, which will result in glowing neurons because the mCherry biomarker that was backwards, once inverted, will now be flipped correctly and expressed. So we will be able to tell that the tool worked because it'll glow red. Implications of this work is that it contributes to future research. For example, any research in the future that requires the silencing of the unc-18 gene can use this tool to do so. This allows that gene to be knocked out so that we can further observe the effects of chemical communication on organismal behavior, and it sets a blueprint for future tools that are meant to knock out other genes entirely. It also, combined with other research findings, can help us gain a better understanding of the association between individual neurons and behaviors, which has applications across multiple species. These are my references. Honestly, I'm not sure what to do about questions, but if you do have any questions, then you can probably email Dr. Rosen and she can pass on my email to you. Or if this is Dr. Rosen listening to this, you could probably just ask me if you have any questions. That's all.